Okay. You guys can see my screen?
Yeah.
Okay. All right. Well, so this lab's going to be a little different than what we've done before. And we're really just kind of thinking through things here a little bit more than physically actually doing something. And I'm going to expand on this a little bit and show you guys kind of what we get when we use the Vesper model. That's actually pretty simplistic. And then if we use like a little bit more sophisticated models as well. So the first thing that we need to talk about is this idea of the lock and key model. And basically the idea is this is kind of has to do with the idea behind drug design, is that there are receptors inside of our body and they have like a little lock on them. And certain molecules are going to have a certain shape that are going to be able to bind onto that receptor. Okay. And that's why we have different kinds of drugs that kind of do similar things, even though they are a little bit different than each other. And each one kind of acts a little bit different to each other. And that's because of how strongly it's binding to these receptors and which different receptors it's able to bind to. Then we get into my little story about ethics here, about opioids and the ethics behind it. Did any of you guys read any of this?
I did not.
Oh, well, it's kind of interesting. So we're talking here about opioids and the fact that these are one of the very few drugs that we have to treat pain. The very first one is morphine. It was isolated from a natural source in the poppy plant and stuff. And then kind of encouraged by the success that morphine had, we started to create derivatives that were similar to it. And some of them were able to get rid of kind of the euphoric effects that led to things like addiction and stuff and treat the pain more effectively. And then we also had people who started to develop things like heroin. And I kind of pulled back from talking about it. I just said the words, what did I say? Unethical researchers. Yeah. And by unethical researchers, I do mean the Bayer company, the aspirin people. It was them. Yep. Yeah. They were hiring Nazis too, like World War II Nazis. So they came out with this guy heroin here, and they marketed it as if it was safer than morphine and less addictive, even though they really knew that that wasn't true at all. So it kind of goes to show you that we really have to vet these things, right? And even the ones like hydrocodone and oxycodone that came out later that were supposed to be better for pain and less addictive and stuff, really what wound up happening is people still got addicted to them. And because they had to take more in order to get the same sort of like effects, it led to this huge opioid crisis that we have now, right? With Purdue Pharma and the Sackler Corporation and stuff. They're being sued right now. And I mean, just a lot of people were very, very hurt or killed by the unethical use of these kind of drugs. So it's important for us to recognize that we really need to vet any kind of drugs that we put out into the world. And it's important for us to actually continue along this research, knowing what we know now, in order to create some kind of drug that could help cure pain, but not have these awful side effects. And that's got to be tested really thoroughly and really well. If we look at this picture here, you can see these two endorphin receptors, right? And this is natural endorphin, and this is heroin. And you'll notice that natural endorphin has this sort of little guy right here, right? That's kind of sitting off there. And what that's doing is that's actually crowding it and making it so it can't bind as effectively to this site. So for that reason, the natural endorphin is going to come in, it's going to bind to this receptor, and it's going to be able to be released. But heroin doesn't have that. And heroin can come in and bind to this receptor really, really tightly and not be released, which is really leading to an unnatural condition, right? And that's why it has the sort of effects that it does. So I fit in a little bit about ethics here. I also start talking about some of the other things that have come and been good with science and been awful, right? I mean, we have nuclear energy and we have atomic bombs, right? We've got the insecticides and fertilizers that help us to grow all the food that we need. I don't know if you guys realize this, but at one time in human history, we were really going to run into a point where there was going to be famine and stuff. I mean, that was somewhere around 3 to 4 billion people. We never, ever would have made it to the 6 to 7 billion people we have now if it hadn't been for the advances that we had in this insecticides and fertilizers that allowed us to grow all the food that we do now. It also led to a lot of the chemical weaponry that's been used in the world. So we really always have to think not only, could we do something? Should we do it? What is the right way to do it? How are we going to protect people from misusing it? And I think that's important. And ethics was something I really wanted to start to work into this course a little bit. So I think we got a little bit of that there. And then we get into part A of the lab. This is about morphine and endorphins, right? And what I did is I provided you guys with this link to this simulator. Have any of you guys taken a look at that link yet?
No.
No? Annalie, did you? You're always on it.
Yeah,
Yeah.
I've been
Okay.
through it a little.
Well, let's click on it and we can talk a little bit about how you use it. So the one that we're interested in here is the one on the left. Okay. So if I click on that, we get something like this. And what we can do is we can add different groups to it in lone pairs and then take a look at what the geometry looks like. So before we get too far into that, let's see what it is we're trying to make. Well, the first question says that morphine and endorphin both contain a coordinating nitrogen, which has 3 single bonds and 1 lone pair of electrons. What is the name of this Vesper model? Okay. So if we look at the picture of, and can you guys see that well?
Yeah.
Yeah. Okay. If we look at the picture here, we're talking about this nitrogen right here, right? He's got a lone pair and he's making this bond back to this ring here. And he's making this bond over to this carbon. And he's got this methyl group here, right? So what does that shape look like? Does anybody have any idea? Uncomfortable silence? Nothing? Okay. So I'm just fussing with that. Let's make it. So we're going to have 3 things bound to it, right? And then we're going to have this lone pair. And what we get here is this shape that is called a tetrahedron. Okay, the tetrahedron is a shape. It's like a platonic solid, which means that all the faces of the tetrahedron are the exact same shape and they're the exact same size. I don't know if you guys ever play like D&D or something, but they're the shapes that they use for dice, right? A cube is a tetrahedron. And that's because when you throw the dice, you're always going to have the same chance of landing on each face. So that is going to be the electronic geometry, right? That means that we're taking into account this lone pair here. But there's also the molecular geometry. And molecular geometry says what is, if we don't take into account this, right? And we just imagine having this shape here, what would it be? And it's a thing that they call trigonal pyramidal. So you imagine that without this here, it would be like a pyramid, a 3-sided pyramid here. Okay. So for fun, what I actually want to do is show you guys a little bit more sophisticated model of what endorphin actually would look like. So our Vesper model is pretty simplistic. It really doesn't take into account all of the interactions in a large molecule like this. This one, I use something called molecular dynamics. And it takes into account all the interactions between the different atoms, but it doesn't take into account the interactions of like the electrons and stuff. That would be like the next step up in our modeling. And you can see here, if we look at this nitrogen, it does have kind of that pyramidal shape to it. Can you guys see that?
Mm-hmm.
But it is kind of flat, right? It's almost flat, right? And the reason is because this guy, in order to get that tucked down in there, right, would get too close to some of these other atoms. So these guys are kind of pushing each other apart while the nitrogen is trying to like push itself down. So you wind up with something that's still got a trigonal pyramidal shape, but it's a little bit flatter than what we would expect with our Vesper model. Let's look at our next question. So what would be the answer to number one then?
Would it be tetrahedron?
It's really both, right? The molecular geometry would be the trigonal pyramidal, and the electronic geometry would be the tetrahedral. Oh, can I turn this off? Oh, yes, I can turn that off. This would be like what you would consider for the molecular geometry. Okay. So both molecules contain a bridge with two carbons that each have 4 single bonds and no lone pairs. What is the name of this Vesper model shape? Okay, so here we're talking about this guy here. This is what they call a bridge because it's kind of bridging together these two carbons here. And this is written in what we would call a very condensed format that you guys will see if you continue on to organic chemistry. So it looks a little odd to you guys at this level. So basically what's happening here is chemists are really super lazy. Okay. So they want to be able to write something that's going to convey what they want, but in the most condensed form possible. So in these models, each one of these lines represent a bond. You can see how they're representing double bonds here. And each one of the vertexes represents a carbon. And each one of those carbons is going to have as many hydrogens as necessary to fill out its valence shell, to make sure that it's forming 4 bonds. So here in this bridge, this guy here is actually a carbon with 2 hydrogens attached to it. And this guy here is a carbon with 2 hydrogens attached to it. So each one's forming these 4 bonds to one another. If we go back to our simulation here, we're in a situation where we have 4 bonds forming. Okay. And we can see that what's happening here is now we have the electronic geometry matching the molecular geometry. And this is something that's always going to happen whenever you don't have one of those lone pairs. Okay. The two kinds of geometries diverge from each other when you have lone pairs, and they wind up being the same when you don't have the lone pairs. So in this case, both the molecular and the electronic geometry wind up being tetrahedral. Another thing I didn't mention, I guess I started to, but the reason this forms a tetrahedron is because this is actually how you're going to get the maximum distance between any one of these, all these guys. This is the furthest away from each other that these can arrange themselves around that central atom. And if you look at the bonds here, you'll see that they're all at 109 degrees, 109.5 degrees. Whereas in the case where that we were just looking at, they wind up being 109 degrees apart from each other here, but it's not showing me. They'll be slightly condensed. They'll be slightly pushed down a little bit in that case, in this case, because this guy's, what they say in the Vesper model is it's occupying more space, even though that's kind of a misnomer, but it's going to kind of push these down a little bit. So if we take a look at our molecular dynamics model, we see that this bridge is right here. And it actually does wind up being kind of a tetrahedron, but you can kind of see how that works with like the building blocks as you go through and starts to fit together. And it kind of winds up warping a little bit. This shape here that looked like a circle before, it actually winds up being kind of this twisted sort of shape here, right? Where you're going up and down and up and down and kind of zigzagging as you go across. And even this guy right here actually wound up not quite zigzagging the way it should and getting kind of a weird tetrahedron shape there. So that's cool. Let's look at our next question. So what was the answer to number 2?
Tetrahedral.
For both, right?
Yeah.
So both molecules contain a benzene ring with 6 carbons that each have 2 single bonds and 1 double bond. What is the name of this Vesper model shape? Let's take a look at this guy. So what did it say that it had?
A benzene ring with 6 carbons and 2 single bonds and 1 double bond.
So 2 single bonds and a double, right? And what we get here is something that is flat now, right? If I look at it from here, I can see that there's just as much above up the center plane as there is below it, right? And if I come up here, I can see that I'm at 120 degrees between each other. There are 360 degrees in a circle. So each one of these is exactly 1/3 of a circle. And that's placing them as far apart from one another as we can get them. If we go to our other model here, we can see that the benzene ring is this guy here, okay? These other rings aren't benzene rings because they don't have that regular pattern like single bond. This is single bond, double bond here. Here we have single bond, double bond. For a benzene ring, you need single bond, double bond, single bond, double bond, single bond, double bond, all the way around. And you see that that is actually kept really rigid in our model, right? It's actually pretty darn flat. It hasn't distorted like any of those other ones that we were talking about. And that's because this is a very stable structure. And those double bonds being stronger bonds are helping to keep this in the shape, maintain the shape that it wants to have. So also, I forgot to mention, this is trigonal planar. Trigonal planar is what we call that. So it's on a single plane, it's a triangle, trigonal planar. Okay. So for number 4, we have both molecules contain 3 oxygens, each with 2 single bonds and 2 lone pairs of electrons. What is the name of this Vesper model? Let's take a look at it. So, Annalie, what are we doing this time? How do I make an oxygen?
With the 2 single bonds and 2 lone pairs.
So it's going to look like this, right? Do we know, can anybody name a molecule that looks like that? This would be the shape that we would have for water, right? This would be a hydrogen and an oxygen here. And you can see that we get this bent in shape here. And that's exactly what it's called, bent. So if I take away the lone pairs, you can see that it's this bent shape. And in practice, experimentally, we're actually going to find this to be less than 109 degrees here. This is going to be smaller than that and kind of squeezed together. Again, because these lone pairs are kind of occupying more space. in our model. Our electronic geometry, though, is still tetrahedral because we have 4 different things, areas of what they call electron density around it. If we go to our other model, we can see that bent shape, right? Even though we're seeing different bond lengths here because we're binding to 2 different kinds of atoms, it's holding that hydrogen a little tighter than it is that carbon. Whereas over here, we have that bent shape. It's a little bit out of plane. You'll notice it's kind of pushed away a little bit. But it has similar bond distances because it's binding to those 2 carbons, and these 2 are similar to each other. And then over here, you can see you have that bent shape, and it actually caused it to bend away. This hydrogen bent away to not interfere with this hydrogen here or this one here. It got as far away from any other atoms as it possibly could. Okay, so that's the end of part A. Do you guys have any questions so far?
So for like number 1 on part A, you could use either option, all right? It could be like trigonal, pyramidal, or tetrahedral.
No, I'd like to see you write both down. Yeah. So I'd like to you guys to show me that you can tell the difference between the molecular shape and the electronic shape.
Okay.
All right. Any other questions? Annalie gave me the thumbs up. All right. So sulfanilamide is a drug that was synthesized in the early 1900s. It blocks the production of folic acid. Mammals don't produce their own folic acid, but instead absorb it from their food. Bacteria, on the other hand, they do produce their own folic acid. So sulfanilamide can therefore be used as an antibiotic. So where if a human or some mammal took sulfanilamide, it's not going to keep us from getting our folic acid because that's not how we're getting our folic acid. It's going to block bacteria from producing their folic acid, and it's going to kill them. So this is why it can be used as an antibiotic. Below, we have the molecule. Consider the molecular representation of sulfanilamide and indicate the Vesper shape for each atomic center. Okay, so let's think about this here. So the good thing about this is we actually don't have to do it for any of the terminal ones at the end, okay? Because they don't fit into our Vesper model, right? A Vesper model talks about having a central atom and it's bound to different groups around it, okay? So there's really, even this looks big, it's not really that many different ones here, okay? So the first one we can look at is this nitrogen right here. What do you guys think would be the Vesper model for the nitrogen?
Three single bonds and an electron pair.
Yeah, but we've got names for it now, right? It's the same as the nitrogen was in the part A.
Trigonal pyramidal.
Right, trigonal pyramidal. Yeah. Yep. So that would be the molecular structure. The electronic one would be tetrahedral, right? Next, we have this sulfur here, right? It's bound to 4 different things. Now we can go back to our model and look at it, but we can see that because it's bound to 4 different things, it's going to be the same as the carbon was before. So what do you guys think the shape around that is?
Tetrahedral.
Tetrahedral, right. And we just talked about the benzene ring, right? All of these carbons are going to have the same shape to them, right? So what was the shape for the carbons?
Trigonal planar.
All right, trigonal planar. And then we've got this nitrogen over here, which is going to have the same sort of Vesper shape to it as this nitrogen over here, right? So we're going to be having the electronic structure of tetrahedral and the molecular structure of trigonal pyramidal for that guy too. So that wasn't so bad. So sulfanilamide is actually not used very widely. Almost very few sulfur containing antibiotics are. And that's because a large percent of the population, somewhere around 40%, is actually allergic to it. And that's true for a lot of sulfur antibiotics. People tend to be allergic to that. It might, however, be possible to mimic the shape of the molecule, but get rid of the problem areas that causes the allergic reactions. One promising method would be to replace the sulfur with its two bonded oxygens. So it wants us to redraw the structure, but replace sulfur with silicon and replace each double bonded oxygen with a single bonded hydrogen. Well, here's the question. Why do we have to replace the double bonded oxygen with the single bonded hydrogen? Anybody know? Okay, I'm getting crickets. So the reason for that would be that sulfur is here, right? And it's able to make those 6 bonds because it has 6 valence electrons, right? So the noble gases have 8, the halogens have 7. Sulfur underneath oxygen is able to make 6 bonds or 2 bonds, depending on how you set it up and whether or not it pairs off those lone pairs. Oxygen is actually not able to make 6 bonds because it's too small. That's kind of a thing that larger atoms are start to be able to do. If we go to silicon, it's below carbon. So just like carbon, silicon can only make 4 bonds. So it would never be able to make the 6 bonds that we see for sulfur, right? It can't make these double bonds above and below it. So we're going to have to replace those with something that's only going to make a single bond with the sulfur. And if we think about what's going to make a single bond, hydrogen we've seen will do that, right? Also, any of these guys, any of the halogens that have these 7 bonds, they can make a single bond. So we're talking fluorine, chlorine, bromine, iodine, antimony. These guys can do that. But they want us to replace it with the hydrogen. So what is that going to look like? I drew it here somewhere. We're going to have this situation here. So now we have a silicon here and we're giving it to these 2 hydrogens, right? And we're going to maintain that same tetrahedral structure around the silicon. So let's take a look at what these guys look like with our other model here. Here we have the sulfanilamide. And you can see what's wound up happening is these 2 oxygens are kind of flattening out here a little bit because they're actually interacting a bit with the nitrogen group here and kind of actually getting a little attracted to it and bringing it over. They're also not, they're also moving out a little bit from this guy here. So they're kind of flattening and moving out in order to get further away from the ring and moving a little bit towards this nitrogen here. The nitrogen is just kind of hanging out there. And you see that it's put its 2 hydrogens upwards here, right? So it's also kind of flattening out that pyramidal shape, right? That we would expect for it to have. It still has that pyramidal shape, but it's flattening it out a little bit. And we would actually expect for this to be what they call a Rotimer Isomer, okay? Where this guy, it had to settle somewhere in the model, but this is actually going to be spinning kind of loosely out in space in the actual world. If we can picture this as a living thing that's moving around, this doesn't have a set place where it wants to sit. It's going to be moving around as this molecule moves through the world. And over here, we see a lot of flattening for this nitrogen, right? Because bringing that down at all would have brought it closer to that ring. It's really almost planar, shoving itself out there to get away from it. But you can still see the effects of that lone lone pair kind of pushing it down below the plane just a little bit. If we go and look at the case where, oh, this is a wrong one. If we go and look at the case where we have hydrogen and silicone, we're getting closer to what we would have actually expected. My model is actually a little twisted here. I don't think this is actually 100% true, that this guy is in the same plane as this guy here. I think this guy would actually be rocking back and forth a bit, and it just happened to settle there. You do see the effect of moving down the periodic table in the aspect that this atomic radius is quite a bit larger, right? So it's a big bulky atom right there. And we can see here that this rotational isomer here, where it kind of moved over to this side, right? That's because this is rocked up here. So in actuality, we're probably going to expect this to be rocking back and forth, and for this guy to be moving around here in response to this rocking in order to keep it away from the ring as it moves along. And it's interesting to note that nothing really changed on this side. This looks very much similar to our other example, because you really don't have to get that far away from a separate part of the molecule in order for the changes to not really change the other structure very much. And this is very important for drug design too. We can change a group, the active group, where something's going to bind or something down here, and still expect that the rest of the molecule is going to have a similar structure. Oh, no, that's right. Okay, so you guys are going to draw me that structure here. And that'll be some good little practice for you guys for when you're doing organic chemistry. Organic chemistry is all about drawing these things out. Does anybody have any questions about part B?
No.
No? Annalie, do I get my thumbs up? Yes. All right. Okay. So now we're on to the tavaborole. Tavaborole is an antifungal drug that is used to treat fungal infections on fingernails. It does so by inhibiting the transfer RNA, thereby preventing the fungus from producing key proteins it needs to survive. Okay. So if you guys studied biology, transfer RNA is what is used for it basically goes and sits on, let me think here, is it messenger RNA? Transfer RNA is the one that attaches to the codons, I believe, down to the amino acids, and then binds to the messenger RNA. So basically what this guy's going to do is it's just this kind of like flat molecule, and it's going to be able to use the shape, its overall shape, to slide into areas in the cell where DNA replication and or protein synthesis is happening. And then the specific shape shapes that it has out here is going to allow it to interfere with that RNA and bind to the RNA. And when you're just trying to stop a process from happening, it's actually pretty easy to do, right? Getting your body to specifically do something is kind of hard. Getting any organism to specifically do something at the molecular level is hard. Messing up something that it's already doing tends to be pretty easy. So that's kind of the action that this guy is going to have. So our question here is, what is the Vesper shape of the boron in the tavaborol? So here is our boron. If we look at our periodic table, we see that it's in this group right here, where it's going to be forming 3 bonds, having 3 valence electrons. So it looks like there's not going to be any lone pairs on there. Who knows what that shape is going to be? Anybody?
Be trigonal planar.
Yep, it's going to be a trigonal planar one. Yep. And it's going to have the same electronic structure as molecular structure, because we do not have any of those lone pairs to affect it there. If we look at our model for that, did I do that one? I sure did. We actually see that what's happening here is we got a pretty twisted up molecule here. And this boron actually doesn't wind up being very flat at all. But that is all due to the stresses of this ring and something that's called ring stress, okay? Because if you look at the different angles that the molecule wants to have, 6-membered rings of carbon are very favorable, okay? You can make a 6-membered ring and you don't have to change the bond angles around those tetrahedrons much at all. However, 5-membered rings just don't work out, and they wind up causing this distortion that we see here. And that's kind of driven this guy way out here. So this is a good example where the Vesper theory really doesn't reproduce the actual shape very well at all. I mean, that's still the answer to our question, because we're only restricting ourselves to the Vesper theory. But it's a good example to show you that why we have to use higher models than the Vesper theory, because it's really limited in a lot of cases. So imagine that boron tavaborol is replaced with a nitrogen atom. What would be the new Vesper shape of this area of the drug? So if we put a nitrogen here, how is that going to be different than when we have the boron? Does anybody know?
The electron pair geometry would still be tetrahedral, but
Right.
the molecular geometry would be trigonal pyramidal.
Exactly, right. So then you're going to have that lone pair in there, and we're really changing the entire geometry around that structure right there, right? We would have a model. Let's see here. We would draw it like this, right? And we're going to have this lone pair here. We're going to have this whole new geometry around here that, like you said, Annalie, is going to have a electronic structure of tetrahedral and a molecular geometry of trigonal pyramidal. I don't think that I did that one. So if we did do it though, we would see that probably a big bending in this shape right here. And I actually might struggle to even get that molecule to converge on here. It's very likely that it either doesn't exist at all because it doesn't lead to a stable structure and it's just good for like a thought experiment for us, or I would have to use some pretty advanced level stuff to be able to actually get it to converge. So I don't think that that's a very likely molecule to be stable at all. And you can see that that would be due to all these stresses, right? So if I put a lone pair right here, it's really going to drive this down. That's going to push it even closer to these hydrogens down here at the bottom. It's going to cause a lot of ring strain and probably wind up breaking one of these bonds, right? If I stress the ring too much, one of these bonds is going to wind up breaking along the way. Okay, so we also have this fluorine down here. And we want to know what's going to happen if we replace it with a bromine. And if we look at our periodic table, we can see that bromine is right below fluorine, right? So it's going to make that one single bond there. What do we think? Is this going to have any effect at all?
I
don't
don't think so.
So
No, it's not really going to change that Vesper geometry at all. It will be larger, right? We saw that in the case of silicon, right? Where when we replace sulfur with silicon, we got a larger guy here that's going to be bulkier. It might push these hydrogens away a little bit, but using the fairly simplistic Vesper model, it's not going to change anything at all. Okay, does anybody have any questions about part C? No? Annalie gave me my thumbs up. So methyl eugenol is an oil extracted from plants. So yeah, so eugenol, there's a lot of eugenol in like cloves. The what do you call that? A spice, right? You guys ever seen cloves before? Usually put it in like pumpkin pie. It's got like a certain characteristic smell to it. And the cloves themselves are really oily. And they used to do something called steam distillation to separate out the oils. And it's like the essential oils that became really popular. And now people put them in little vaporizers and, you know, make their houses smell nice with them and stuff. That's methyl eugenol is one of those essential oils. So it's been used extensively as an additive in foods. It's used in perfumes and aromatherapy oils. There's also a popular homeopathic salve for treating arthritis. So homeopathic, typically means it doesn't really work, but you know. Let's look at this guy here. So we got the two oxygens have the same shape. What is the Vesper shape name? So what do we think that the Vesper shape name of this oxygen is going to be? How many bonds is that forming? How many lone pairs does it have?
Is it forming two bonds?
So it's forming two bonds. And it's got two lone pairs, right? If we go back to our periodic table here, this is 8, 7, 6, right? So you have the two lone pairs that takes up 4 of those. You have 2 electrons left to share in a covalent bond. So this guy's got 2 lone pairs on it. It would look like this if I had written it out with the lone pairs on there. So what is that Vesper shape going to be? We already did oxygen, so it should be pretty obvious.
It'd be bent.
It's going to be bent for its molecular structure. It's going to be,
Tetrahedral for-
again, yeah, that's right. Tetrahedral for its electronic structure. Let's see. Yep. Now here, right, in the interest of looking at this in a condensed fashion, we drew these two pointing towards each other here, right? But in actuality, in our actual model, they moved away from each other in order to be further away from each other, right? And you can see that when we write it with this condensed format, a CH3 looks like just a little guy there. But in fact, that's a pretty bulky group, right? If these guys were both bent into each other over here, they'd be like overlapping with one another. So they move away from each other in our actual model. And then this guy over here, again, we have this rotor isomer here situation, right? Where this guy has gone to this side of the plane, but there's nothing special about it due to the symmetry, right? This guy could just as easily be on the other side here. And in actuality, if you were to lock it in and look at any one of these molecules, you're going to see this kind of turning and moving from one side to the other in actual space, because our molecules aren't completely static things. And again, we also see that this benzene ring kept really, really rigid, right? It's got that nice flat shape, did not change much at all. Benzene rings are well represented on a flat piece of paper. So the last thing we have here is says circled the methyl eugenol analogs that theoretically could have the same effect due to them having the same shape as the compound above. So we're replacing the oxygen in each one of these. What do you guys think would be the best one to replace it with? What do you think? What do you think we're looking to replace it with in these cases? Nothing? Okay. So what we want to do is we want to replace it with something that's in the same group as the oxygen, because that's going to lead us to that same sort of shape. Okay, so we want to replace it with like a sulfur or selenium or something like that. So if we want that to happen to keep that bent shape, well, here this guy isn't going to really do it, right? It's going to have like a kind of a weird shape there. But here where we have sulfur and selenium, this would be a viable analog. Here where we have oxygen and sulfur, that would be a viable analog. I don't think I see another one that's going to be a viable analog for us. Does that kind of make sense?
Yeah.